This thing is like not completely flat.
Okay.
You don't have like tape or anything to like strap it down?
I won't touch it.
Yeah. Again, I'm gonna start kind of broad, like the last time, so you guys could see kind of where everything is coming from, right? So I'm gonna start with glucose. This one is red as well. And as we all know, glycolysis produces pyruvate, right? And then from pyruvate we can make acetyl-CoA through the pyruvate dehydrogenase complex, right? And then there's certain things that we can do with this acetyl-CoA, right? One of the things we can do is ketone bodies through ketoneogenesis. We can, as we all saw last week, right? We can make malonyl-CoA to produce
Fatty acid.
fatty acids. And now we know that we can also take this acetyl-CoA and make cholesterol. And so that's one of the things that we can look at, right? Cholesterol synthesis, how it's made, and like I said, not too much of the regulation, but some of the implications of cholesterol buildup rate and some of the targets for specific medicines with that, like statins, right? So we'll go over that. Okay. So we're gonna start out with two molecules of acetyl-CoA, right? We're gonna fuse them together, and we're gonna make acetyl-CoA. And CoA is gonna be.
So part 3 is just the cholesterol synthesis.
So we take two acetyl-CoA molecules, we make acetyl-acyl-CoA, right? Do you guys remember how many carbons is each acetyl-CoA? Two, right? So we make this 4 carbon. I'm gonna number the carbons, right? So that's gonna be kind of important. And then we're gonna take another acetyl-CoA, and we're gonna produce something that's called HMG-CoA. And the enzyme that catalyzes this reaction is HMG-CoA synthase, right? The making of HMG-CoA synthase. Okay. And HMG-CoA synthase is, we added 2 carbons, it started with 2 carbons, we made a 4 carbon, we added 2 more carbons, so it's a 6 carbon intermediate cholesterol. And then HMG-CoA is going to get reduced, and this is the most important step because it's the committed step, and it's going to be the step that is going to be subject to regulation, right? So we're going to make valinate. And you guys remember the name of this enzyme? HMG-CoA reductase. So basically HMG is getting reduced, and remember, whenever we have a reduction process, what is the molecule that's going to be providing our hydrogens?
LDH.
It's converted to NAD+. The valinate then undergoes a series of steps that produce what is called isoprenes, right? And the ones that we do need to know. I'll go over those. So valinate, a multi-step process, which basically what happens is the valinate is a 6 carbon, right? We're gonna produce this 5 carbon isoprenes. So we undergo a decarboxylation. I'm not gonna go over the details because that's not one of the intermediates that he was gonna test us on. So it undergoes a decarboxylation, and we basically create this molecule called pyrophosphate, which essentially has 3 phosphate molecules, So we're gonna invest 3 ATP into creating this 5-carbon isoprenes. Okay, so we'll talk about also I think that was probably in the quiz, it might not be here, but just briefly about energy equivalence. You guys remember he asked us in the homework and in the quizzes how many, let's say, ATP molecules or acetyl-CoA molecules takes to make a specific intermediate or cholesterol, right? So for every we know that for every 5 carbon, right? For every 5 carbon, we are needing a total of 3 ATP. So of course, cholesterol being how many carbons? 27, right? Well, yeah, we can say 30, right? Because that's the last intermediate. For 30 carbon, we're gonna need a total of 18 ATP, right? And same thing for acetyl-CoA. For every 5 carbon of acetyl-CoA, we make we need 3. For every 5 carbon we need 3 acetyl-CoA. That was one of the quiz questions, right?
Yeah.
And for every 30, of course, we're gonna need 18.
Those were both 18, right?
Yes, so
I
they're both
haven't
18.
even checked my quiz.
Yeah, so there's for mine, I don't know if it was different for you guys, but it was like to get to geranyl what is it called?
Geranyl?
Yeah,
Geranyl.
which is a
Geranyl
10
or
carbon intermediate. Yeah, you needed 6, right? Because it's 10 carbons. You're in 3. So just know that from here to create isoprenes, we need an input of 3 ATP. So that's kind of where it comes from, right? So the activated isoprenes that we're concerned with is, I think you wanted us to know maybe it's the isopentyl pyrophosphate, right? And dimethylallyl pyrophosphate. Okay. And if you take a look at the names, it kind of tells you like basically 1, 2, 3, 4, 5. So iso basically means that the double bond is here, and for dimethyl, it creates 2 methyl groups, right? So the double bond is kind of in the middle of the molecule. And then you have basically, I'm just gonna do this, 2 phosphate groups. Pyrophosphate means 2 phosphate groups. So those are how our isoprenes look like, right? Do you guys remember from the slides? It's like basically a long version of that, right? Because you keep adding 5 at a time. So it looks like something like crazy like that, right? A bunch of carbon, hydrogens or hydrocarbons arranged. And then from these, so we have 5 carbons, right? From these, like I said, it's just gonna be a matter of adding 5 carbons at a time to create all these intermediates and eventually to get to cholesterol. Okay. So then we're gonna go through those reactions. So whenever you add another isopentyl pyrophosphate, we're gonna create the intermediate that we need to know is geranyl, right? And that is, we're adding a 5 carbon, another 5 carbon, so that's a 10 carbon intermediate that we need to know. And the mechanism or the method, right, is called head to tail. Basically, you're taking 2 of these, right? And instead, this would be basically the tail, actually, the head. And this would be the tail, right? If you were to add them together, you would add them in a head to tail kind of manner. So that's what it's referring to. It's just the way that they are added in which side. I think there was a quiz or a homework question like that.
It was like to like number them or something.
Yes.
I totally like messed that one up.
Okay. And then we're gonna add another 5-carbon isopentyl pyrophosphate, right? Another into that to make a 15-carbon, which is it's farnesyl, right?
Farnesyl, yeah.
Farnesyl. Which again is a 15-carbon. So we're basically adding another isopentyl pyrophosphate. And again, this is going to be a head to tail mode of adding the carbon. And then from farnesyl, so we have a 15-carbon, right? We're gonna add another 15-carbon, and that's gonna create a 30-carbon. So we add another farnesyl molecule right to this. So, and this one is a head-to-head mode of addition. Head to head. So the first two, head to tail, head to tail. This one is head to head. We're just joining basically those carbons up together. And this one is gonna make squalene 2,3 epoxide. Actually, we don't need to know, right? Just make squalene. Yeah, I'm gonna do squalene. Squalene 2,3 epoxide was probably relevant during the homework when he asked what is the last common intermediate between fungi and plants, right? And it would be the squalene 2,3 epoxide, but for this, for the exam, it doesn't look like we need to know it. So squalene then is a 30-carbon molecule, right? We added a 15-carbon to another 15-carbon. Okay. And then squalene, and I'm gonna put this as multi-step because it takes multi-step. If it takes a few other steps, it's not just one. And then squalene gets turned to Lanosterol.
That was like one of the answers of the quiz too, right?
So the difference is basically Lanosterol already has and this is a multi-step too, I believe. The oxygen group on it. So if you take a look at cholesterol, right? We have 2 6-carbon rings and 1 5-carbon, right? 1 5-carbon ring. A, B, C, D. And then we have something that looks like this. This one here. It looks something like that. But I might have added a little bit too many carbons, right? But it 1, 2, 3. At the position number 3, it has an OH group, right? And it has a double bond between here. So to know, some things to note about the structure of cholesterol. Again, it's a 27-carbon. 4 rings. And it has an OH or hydroxyl alcohol group at position number 3, carbon number So if you were to look at lanosterol, it basically has already the oxygen group on the third position. So that's one of the things to note. I think that was also in the quiz. It was like which intermediate has 30 carbons and it has the oxygen group at position number 3. That would be lanosterol. So 30 carbon intermediate with oxygen at C3. He might have just thrown in another question like that. And this is cholesterol. And then again, series of multi-steps, right? We go from a 30 carbon to a 27 carbon, which is what, you know, the product that we want, and that's cholesterol.
Is that lano? What does that say?
Lanosterol?
Oh, no. Thank you.
Yes. Says 27C. You guys good so far? Any questions? Yeah, so just take a note of those intermediates, right? Mevalonate, 6 carbon, the isoprenes, the 5 carbon, geranol 10 carbon, farnesyl 15, squalene 30 carbon, and lanosterol 30 carbon with oxygen at C3.
The cholesterol also has the OH, right?
Correct. This is the structure of cholesterol. At C3.
The only enzyme that he specifies is the HMG.
Reductase, right, because that's the one that is going to be subject to our statin therapy. Basically, whenever you have high amounts of cholesterol, right, called hypercholesterolemia, this is going to be the subject of inhibition, right? So this is, like I said, your committed step. Write that down. So HMG-CoA turning into mevalonate is our committed rate limiting step of cholesterol synthesis, right? So statins, and this was the question that you had, Christina, they resemble HMG-CoA. They resemble the molecule, so they work by competitively binding to the enzyme, right, HMG reductase, that then doesn't allow for mevalonate to form. It's essentially, you know, kind of truncating this pathway, so inhibiting. So it's statins inhibit MG-CoA reductase through competitive inhibition. Basically, the statin or the actual medication is similar to, looks like, HMG-CoA, which is the substrate of that reaction.
Oh, that was the answer.
Mm-hmm.
I put aspirin. I mean, I remembered the aspirin part, like in the whole thing, but I didn't remember the HGM.
So aspirin is for
like the COX and all
yes.
that stuff, but like, you know, cause I was just like
Cyclophosphamide.
that, like, you know, it was around the same, it was the same type of thing, and I was like,
It works as, like, if you think about it, that's generally how most medicines work. They, you know, inhibit, they look like certain substrates of certain enzymes, right? And they inhibit that enzyme, either by competitive, and if you guys happen to remember biochem, there's like non-competitive, different kinds of inhibition, right? And of course, this one in particular inhibits the committed rate step, right? And basically inhibits the entire process, which is good, right? You know, that's what we want for people with that have high cholesterol that tends to lead to things like cardiovascular diseases, right? To be able to stop this process from creating too much cholesterol. So if anything, know this step, know this substrate enzyme product.
So that's what I like. That's where I get confused. Like I know that ACE is always like an enzyme, but that's why I get confused of which one it is, or like you know.
Yeah. So MGA-CoA reductase is your enzyme.
That's
It's basically
the one. Okay.
yes, it's reducing the MGA-CoA, which is the substrate, and the product is mevalin. I think if you think about it like that, it's product or substrate enzyme product. Right?
So that's say it again. Sorry.
So this will be the substrate, right?
The substrate is
Yes.
that one. Okay.
Basically, what it's taking,
Yeah.
what the enzyme takes to convert, your product.
Okay, it's because when I said substrate, I thought like something that works with the enzyme, so I put NADPH.
Oh no, that's coenzyme.
Coenzyme, okay.
Yes.
But it's that's not even a coenzyme either.
No, it's actually I think it. Would you consider a coenzyme or an actual
Is it
reactant?
a substrate? It's like a
I
reactant.
think it would be because it's actually
I think if you write out the equation.
it might be a coenzyme because if you guys think about all the ones that we have learned before, like biotin, tetrahydrofolate, they're actually providing something, right? They're providing more than likely, like usually it's gonna be a hydrogen, right? Or there's other ones like they provide a methyl group. So I think it's safe to say it's a coenzyme, but I can ask him, like.
No, if it's not important, then it's okay.
No,
but
no,
now I
just
know
know-
substrate is like the product that you had before.
Exactly.
Okay, that's
exactly.
all I
It's
wanted.
what the enzyme's gonna work on. If you
Okay.
think about it.
Yeah.
It's working
Now
on this to create this, right? Kind of thing.
Okay. And the PowerPoint, they just not give it malavonoid. Looks like malavonoid. That's just
I think that's possibly what the end product looks like. Maybe you kind of saw all those like rings and everything. Maybe that's where it stops. That's the product that it creates, right? Instead of mevalonate, it creates like mevalonate something else, right? And then the body gets rid of it. And you basically the main thing to know is that it inhibits the formation of cholesterol to reduce the amount or basically the concentration of it. Pretty much throughout the body, right? That can be problematic. And then of course we know that cholesterol can make different things. Let's go over that just in case he throws in like another bonus question like what he did. Cholesterol is useful for different things. So with cholesterol we can make vitamin D.
I was so mad I got that question wrong on the quiz.
No, I knew that one. I just didn't know the other one.
It
The-
makes which one?
I got the lancerol. I think it was the one of the squalene or something. I
Squalene.
haven't checked it yet,
Okay.
but I think it was the one of the squalene or something. That was one of them, right?
Yeah, I think so. It was squalene and I think.
And then I mixed up the order of those. So then
okay.
it's the isopentyl
Mhm.
pyrophosphate, and then it was the journal. Then it goes to the fernal. So, and then, but there was 5. 1, 2, 3,
It was
4.
Mevalonate.
yeah, so
Mevalonate.
if you were to number them, right, it was mevalonate,
That's what I named
isopentyl
too.
pyrophosphate, geranyl, farnesyl, and
Because who knows, maybe he'll just gonna copy and paste the same things.
yeah.
I
So
think-
make sure you.
Yeah. I think I switched 3 and 4
you
too.
might switch.
I think.
Okay, and so cholesterol can also make something that's called cholesterol esters, right? Cholesterol esters, and basically what cholesterol esters are, you still have the same composition of 4 rings, but it just has a fatty acid. So let's draw that. So you have this.
It has the fatty acid instead of the OH?
Yes.
Or, okay.
Let me show you how that looks like.
Yeah.
So you have these 3 6-ring carbons, right? And then you have your 5 ring carbon, and then you have looks something like this. I might have added too many carbons, but
It's okay.
here it will have this. You have that O, right? You to fatty acid, like this. And this is your cholesterol esters. And then cholesterol can also make steroids, right? It can make estrogen, progesterone, testosterone.
I wonder if you'll put that.
Maybe it's a bonus.
Maybe, yeah, it's true.
Yeah, maybe if it's a bonus, so just note. Okay. Are you guys good with this? You guys good with the order and everything?
I know, and you're like, be like,
I would review maybe also, because you know how the last one of the quizzes he had like the actual molecule, like how they look like, and like it's like he would just show you, okay, this happens, right? What is the enzyme? So just review.
Like for the fatty acid synthesis?
Yes, I think he had like a reductase.
The last
on there.
step.
Yeah. So take a note about how they look like, right? You'll know like it's a 6 carbon. If it has more hydrogens, right, it's gonna be reduced, right? But you have little things.
he
to-
put both of the quizzes on there, like with answers
What do you mean?
for both of them, the 3 and the 4.
Oh, he already.
They should be
I-
up already, yeah.
Yeah, he posted. We could see them
I
already.
think it was after mine were open, because mine opened up at 3.
Oh, okay. Oh, yeah, that's right. You still
Yeah,
had to
because I
take it.
had my extension.
Yeah, that's good that he gave you a chance.
Yeah, I know. I was so grateful. I was like, oh, thank you. He had an answer too. I woke up and I was like, oh my god, please. I just need the extension.
I think he'll only
I'm glad
give
he
you one
gave it to
chance, and
you.
then if you
No,
said
yeah,
the
for
pasta,
sure. Like imagine
mine
if I was still
passed.
like, hey sir, like.
I know mine passed, and I'm like, because I was working on the homework that day. So I was like,
I'm glad he gave us an extension on the homework too, because that homework, both of these last homeworks are due tonight.
I already did them all.
Yeah,
I
I
got them all
finished them
done.
today. I
Like
finished
I
them
was like
earlier.
working on them all, and like I need to get them done because it's gonna help study.
Yeah.
Only vitamin D is the only vitamin that's made out of cholesterol.
I think so too.
Yeah.
And then also bile acids. That's really important.
Oh yeah, bioluminescence. That one like messed me up on the on the homework or something like that.
Okay.
And what was the answer to that one?
Was it the one that?
I'll check right now.
It was how do bile acids affect
Oh.
or
I think it was how they're acted upon by sequestrants.
yeah.
So we'll go over that. But yeah.
Yeah, we should.
Yeah, we'll go over this. Maybe he'll throw in a bonus, or are they actually part of the? Actually, it is part of the review, so we'll go over that. That's why I drew the diagram over there. Okay, and then it's like, okay, well, we made cholesterol, like, how do we transport it and everything, right? So we have different lipoproteins, and what lipoproteins is just how, or the way for the body to transport very hydrophobic molecules, right, such as cholesterol. If you take a look at it, there's only like pretty much this OH group that has some polarity, right? Everything else pretty hydrophobic in nature. So this is the way that we're going to transport them throughout the body, and we have 4 different kinds of lipoproteins that he might quiz us on, right? So we have HDL, high density lipoprotein, right? We have LDL, low density lipoprotein. We have VLDL, very low density lipoprotein, and chylomicrons. And I have
Is the HDL.
Of course, he's like, oh
Biggest
no.
is the chylomicron. Yeah, like at least we'll for sure get this one right. Come on.
Okay. So, and they transport different things, right? Your HDL and LDL are going to be your main transporters of cholesterol. So cholesterol. And your VLDL and chylomicrons are going to be your triacylglycerols and cholesterol esters, right? Triacylglycerols. I think actually your VLDL is the ones that transport the highest portion of cholesterol esters.
So they both transport triacylglycerol, but the VLDL also transports the
Cholesterol.
cholesterol.
Yes, and it also depends, where that transport, how that transport flows, right? Basically, HDL is considered the good cholesterol, right? Because it transports the cholesterol from the tissues to the liver. While LDL is considered your bad cholesterol, right? Because it's the reverse of that. From the liver to the tissues.
Yeah,
It's
that's why-
basically-
what was that, but what was the answer of it?
It's
was
that
a
a defective.
A
Yeah.
defective LDL receptor.
So what happens is, if the cell isn't able to take up LDLs, of course they're gonna build up, right? So when this mechanism or this endocytosis can't happen, right? Because of defective receptor, then what happens is LDL builds up in the bloodstream, right? Because the cells aren't able to take it up. And so when that happens, that can lead to a lot of problems like atherosclerosis, which is basically LDLs kind of sticking to the walls of arteries, right? And causing like plagues, right? So that we can go over that, right? So coronary artery disease, it's basically the buildup of LDL is correlated to high levels of LDL in the bloodstream. It has a positive correlation. So if you think about it, this will be your LDL, right? Concentration. And this is your, I would say your, what is it called? Incidence of heart disease, right? Of COD, coronary artery disease. It's a positive correlation. The more LDL found in the bloodstream, the higher the incidence of coronary artery disease. So, and that happens because you have faulty receptors, faulty LDL receptors. And then another thing that also can be a risk factor of CAD. Let me pull up the slide here. If you have low levels of HDL, if you guys remember, your HDL is your good cholesterol. It's what picks up cholesterol from all over your body and brings it back to the liver. And that's good, right? Because we want that cholesterol repurposed to create bile acids, to create steroids. So if your body is not capable of bringing back those cholesterol molecules because you have low HDL, then that's going to be a problem, right? And that also is indicative of CAD. So the low levels, it has kind of like a negative correlation, right? The lower the levels of HDL, the higher the possibility for CAD, right? So this would be like a negative correlation. You have HDL here. The higher the levels, the lower, the levels, the more of an incidence of coronary artery disease. Does that make sense? It kind of makes sense, right? Like basically the transport of it can have a lot of repercussions, whether the cells can't take it up or you can't transport it out because lack of HDL, right?
Yeah.
It's gonna have problems with the blood vessels are gonna have problems with building up of the cholesterol, causing again atherosclerotic plaques.
So it's just too much of the bad and too little of the good.
Exactly. Too much of the bad, too little of the good, too much of the bad. I think he's gonna ask a question about this.
Yeah.
He put on there coronary artery disease. So now we know also that cholesterol can be repurposed to make bile acids. And again, bile acids are important for digestion, right? Put here. Acids aid in digestion, and they are basically emulsifiers. Right? What that means is basically all those fatty acids that you consume, or those or cholesterol, triglycerols, the diet, the bile acids help to kind of like emulsify them, which basically is like kind of like shaking, right? Because they're very hydrophobic, so it's a way for them to kind of like break them down into their components. And so now we can go over a therapy for also combined with an inhibitor of HMG-CoA, as you guys know, statins, right? If you combine statins with sequestrants, bile acid sequestrants, your cholesterol levels are going to go down even more, right? So this is basically a way to repurpose bile. Since bile basically is recycled, it can, so if you guys know, bile is produced in the liver. This is your bile, right? Produced in the liver, and then it comes down and it gets stored in the gallbladder, and then the gallbladder secretes it from the bile duct to the intestine, right? It's like the duodenum, or whatever. And then what happens is they go through digestion and everything, but the bile gets recycled back to the liver. But what happens with these sequestrants is they bind to the bile acids. So here comes your little sequestrants, right? And they're gonna bind like this. And what happens is instead of the bile being recycled, it's just gonna get excreted. So what do you think happens when bile gets excreted rather than recycled? That's basically going to reorient cholesterol, too much cholesterol that has been building up, to make more bile acid. So instead of it being circulated or whatever, it's gonna be repurposed to make more bile acid. So it's a way to kind of keep the concentration of cholesterol low, right? Because the body is basically excreting all the bile it's making, so it's like we need more. Let's repurpose the cholesterol that we have to make more bile. So it's just basically redirecting the creation of cholesterol to make more bile, keeping it out of the bloodstream, basically. Does that make sense?